Hi, this is Dr. Yunlong Liu. In this short video, I'm going to give a very brief introduction about the single-cell analytics. I want to answer several questions. Why, when, how, what to expect, and what is next. First question, why we do single-cell analysis? We know that most of the tissue or organ in our body are complicated, are composed of many different cell types. In this one example that we did for single nucleus RNA sequencing on human frozen samples, as you can see that out of 1,500 nuclei we sequenced, and they are coming from many different cell types: astrocytes, neurons, microglia, oligodendrocytes, and OPC. And actually, there are many other cell types as well. So after getting this data, we think a little bit more about the RNA sequencing, the bulk RNA sequencing we have done in the past. What are we really measuring? It is actually a mixture of all these different cell types. And sometimes if we see the differential expression of certain genes, and that simply can come from the proportion differences of different cell types. Or in some other cases, even when we are interested in neuron, for example, and some genes differential expressed, we may not be able to detect those differential expression just because they are mixing with many different cell types. And in that case, that doing single cell analysis will greatly help us to increase the sensitivity of the measurements. Single cell will help us to unravel the heterogeneity. So we understand that in tissue, that for example, if we do cancer study, that the tumor we got is not pure tumor cells, and it will include many different tumor subclones as well as cells from microenvironment and many immune components as well. I just give you an example of brain. They are composed of many different cell types. Sometimes when we study developmental study, for example, if we study ES cell or iPS cell, and we understand that in our Petri dish, actually there is a mixture of many different cell types at different stages or different lineages, and doing the single cell analysis will help us to understand the biology behind it. And sometimes even in the cell line studies, there are many studies reported there are variabilities and coordination among cell types. Using single cell technology will help us to understand those mechanisms. To decide whether you need to do single cell analysis, and the first thing you need to consider is, does your research require single cell sequencing? In a short word, is whether the heterogeneity of the different cells are important to your study. And if it does, probably single cell is a good option for you. The second thing you need to consider is the technical feasibility. For example, are your cells alive? And in most cases, we'll prefer to have cells that are healthy and very much alive. In that case, that the data will be in a high quality. But we also understand it's not always possible. If it is, that whether there are some other ways that we can deal with it. For example, I showed you the fresh frozen, the nuclei sequencing on the fresh frozen tissues. And the second question is, do you have a good protocol to deassociate the cells? And what are the size of your cell? Do you have enough cells? And sometimes we do need to have thousands of cells or 10,000 of cells. And does your cell prep have a lot of debris? If it does, you may need to think of a way to clean your cells. And in that case, that potentially can increase the loss of the cell and increase the stress you impose to the cells. And the last question is whether you have enough funds. We all know that these experiments are not cheap, and you do need to consider those cost issues in your study designs. The next question is, how do we do single-cell analysis? It includes several different steps: single-cell capture, cell lysis, capture mRNA, library construction, and sequencing. And most of the innovation in recent years occurred in the first level, which is the single-cell capture. On the left side of this figure, it shows you several low-throughput technologies, but most of high-throughput technology today is a micro droplet-based approach, as well as a microfluidic-based approach. In our institution, we do have access to the 10x Chromium systems, and this figure shows you how that will work. And I'm sure there are a lot of YouTube videos which I will pull out and then hopefully that put into our playlist. But generally, and this is what the principle. So you start with this tube. In this tube, there are many 10x barcode gel beads. And if we zoom in for each gel beads, and they have a lot of oligonucleotide that is attached to it. If we put one out, that will have R1 Illumina adapters sequence, 10x barcode, and this is a short nucleotide sequence that is different for across all different gel beads. And this is this primary sequence that we are going to use to decipher which molecule coming from which cell. And we also have UMI as well as excuse me, the poly-T sequences, and this will be used for the reverse transcription. So before we do the experiment, we push this 10x barcode beads into this channel. And this channel will come in with the cells. So you can see that if we control the speed correct, and at a certain point, that the cell will meet with the gel bead. And we try to control this, that do not allow that two cells to meet with one gel bead. That will cause doublets. In that case, we will not be able to do the single cell analysis. So after this procedure are being conducted, and we'll push this part into an oil, we know that when the water molecule coming into the oil, they will form all these bubbles and isolate the bubbles, which means the future reactions will all be contained within this bubble. And we'll collect these bubbles, and you can see that many bubbles have the cell and the gel, that they are being fixed together. And then we'll do the reverse transcription that occur within these bubbles. And as you recall that the gels have the barcode that will specifically tell us which nucleotide this particular gel coming from, this oligo come from, and all the molecules in this particular bubble will be barcoded by a specific barcode. And after doing that, we can remove the oil and they can be mixed, and then we can do the sequencing. So when we do the sequencing, we will first sequence the barcode side, and to make sure we understand which cell this particular molecule coming from, and then we will sequence the other side, which is the gene side, and then we will know that which gene it come from. So in this case, we'll be able to sequence doing the single nucleus or single cell RNA sequencing. How to do the experiment? And the first case, which is the most common case, is we will do one individual samples at a time. So you bring your cells and you get the single cell suspension, and then we will load into the machine, and then procedure will go on. And in that case, that we can sequence up to 10,000 cells per sample. Sometimes when the funds are tight, that people are inventing in the way to multiplex samples. And I have to put a precaution on this. This only works for human samples and for when you deal with many different human individuals, because when we finish the experiment, we want to informatically decide which cell coming from which individual based on their genetic variants. If you are using cell lines, for example, or animal models, for example, all the cells coming from the same have the same genetic background, we will not be able to distinguish which cell coming from which individual. So, however, that if you work on the human cells, that they all different individuals have different genetic variants sequences, and we will be able to informatically decide which cell coming from which individual in the analysis stage. So there is a very interesting paper I encourage you to take a look at if you're interested in doing this type of experiment. And the next part is we can also do the multi-sample analysis using hashtagging approach. So the general idea is you bring your cells from different samples. In this case, we are having the cells coming from 3 different samples. Before we mix them together doing single cell analysis, you actually incubate your cells with a generic antibody that carries a cell barcode. So you can see this one is blue, this one is purple, this one is green. They all carry different nucleotide sequences. In this case, all the molecules or most of the molecules that some of the molecules coming from this cell will carry this green barcode. So in that case, we'll be able to mix all the samples together and then do the single cell analysis. Eventually, we will decide that which cell coming from which individual by seeing that whether some of the sequences is carrying a specific sample barcode. So there are very established approach to do that, and our sequencing center support this type of experiment. So I'm showing you here is the example we did using the second approach, which is coming from the 10 frozen human samples, brain samples. And we actually mixed them together and then did analysis. And I can tell you that we are able to de-identify individual cells and nucleotide, which sample they come from. So in this case, we mix 10 brains together. We superload the system, generate 42,000 cells across 2 wells of 10x machine. And then by the end of the day, that we spend about $6,000 and get 10 brain nuclei sequencing done. So by average, it's only about $600 per sample. The next part I want to go through a little bit is what can we do using the single cell analysis or 10x machine that we do have. So the 10x system currently supports several different applications: single cell, single nucleus RNA sequencing, single cell ATAC-seq, single cell copy number variation, and single cell VDJ segmentation analysis for the immune systems. And we also support CytSeq or RIP-seq or 10x feature barcode. And these assays will allow you to quantify the RNA level as well as cell surface proteins in the same cell at the same time. So that is a very important application that is being desired by many different analyses. So I also encourage people to take a look at the future assay development. So there are many development in recent years. And for example, the 10x just announced by the end of the year, they will start to support the protocol to do the single cell RNA sequencing and the ATAC-seq from the same cells. And that will be a very important application for many groups. The next question I want to answer is what to expect in terms of analysis. I'm giving a very, very simple example. In left case, there is a healthy sample, and in the right side, there is a pathological sample. And when we do the single cell analysis, there are three levels of information that we can understand. The first one is within individual cell types, whether there are transcriptional variability. Of course, you can study regulatory network and the transcriptional activity for the additional informatics analysis. The second question you can answer is what are the genes that differentially express between different cell types? And there are a lot of different analysis will support that. And the third one is whether there are differences between two different conditions, both in the proportion differences of the cell types or cell type composition, or within each cell type, whether there is a differential gene expression. And if you do the analysis in a careful fashion, all these questions, you can fairly address those. For stem cell studies, and there is a possibility to study the single cell trajectory as well, and including the cell when we do the cell development. So I'm listed two different tools here. On the left side, it's called monocle, and the right side is called RNA velocity. And both tools are very well done, and you can. So we do have a short module to demo how to use monocle in the later slides. This is one of the examples that we have done for the single cell SCATAC-seq. So you can see this is on the multiple myeloma patient samples. You can see there are many different cell types are being identified. But more importantly, that when we look at this, so you can see that this is the relative positions of the signal away from transcription starting site. We can see a strong enrichment. This tells us that this is good data for you. That is what you are going to expect from a SCATAC-seq. And also that in terms of fragment sizes, you can see that this is a nucleosome-free region. This is one region that cross one nucleosome, cross two nucleosomes. So you can see that this is give you a good indication. This is a good quality of the data. To do this type of analysis, this is one of the examples we did for single cell SCATAC-seq from the human brain samples. So you can see we are able to identify single cell or cell type specific chromatin accessibility from different cell types. And this is a very important information for us to understand. After single cell analysis, and many different groups are starting to think beyond the single cell analysis. One of the caveats of single cell analysis is the spatial information are being lost, because when you get the tumor, get the tissue, get the brain samples, and when you do the single cell suspension preparation or the single nuclei preparation, and the cells are all mixed together. So the spatial information are being destroyed. In many cases, we know that those spatial information are super, super important. And that there are several different technologies that people are using to recover the spatial information, including the 10x IUSM system or this codex system for the protein profiling, and the single molecule FISH or imaging-based transcriptome profiling. And that's all I want to talk about today. So in this video series, we're going to give you some of the demos of how to use different type of tools to do this type of analysis. I hope you enjoyed. I will see you this Friday.